Hello everyone, welcome back to my YouTube channel. Today I am going to teach you the innate immune cells produce the molecules responsible for the type 1 hypersensitivity symptoms. So when the innate immune cells produce the molecules from the cells, when mast cells, basophils and eosinophils react by linking FcεRI to IgE antibody, and second, exposure of allergens binds to the antibody, which I discussed in my previous video. A link given into the description box, so please watch it. And then once they binds with the FcεRI to IgE, and when second exposure of antigen that binds to the antibody, further they release the molecules that cause the symptoms of type 1 hypersensitivity. There are two types of mediators which are produced by the innate immune cells. One is the primary and second is secondary substances. Okay, so both have the effect nearby tissue and other cell like the eosinophils, neutrophils, T lymphocytes and monocytes and platelets. Now, when the body encounters parasitic infection, it releases helpful substances that trigger defense mechanism. These are substances. Okay, this includes widening blood vessels, making them more permeable, allowing more plasma and inflammatory cells to target the invader. So all of these substances also directly harm the parasite. Now, on the other hand, when allergen causes the release of these substances, it leads to unnecessary increase in blood vessels permeability and inflammation, and that causing the damaging to tissue without much benefits. Okay, also that is the damaging to tissue. Now we know the types of molecules. Okay, we can say type of molecular mediators, okay, produced by the innate immune cells. So primary mediator first are performed and stored in granule prior to cell activation. Okay, whereas secondary mediators are either synthesized after target cell activation or released by the breakdown of membrane phospholipid during the degranulation process. So when the degranulation process, after they release the secondary mediators. Now here in this table you can see here mediators. Okay, these are the mediators all effects. There are primary, that means these all are primary mediators and these all are the secondary mediators. So first we discuss the primary mediators. The most significant primary mediators are histamine and proteases, eosinophil chemotactic factor A, and neutrophil chemotactic factor, and another that is a heparin. Okay, serotonin, which is present in rodents. Okay, so first we understand the histamine. Okay, how histamine formed. So which is formed by decarboxylation of histidine amino acid. It's a major components of mast cell granule. I think that is approximately 10% of granule base. Okay. Its biological effects are observed within minutes of mast cell activation. Once released from mast cells, histamine binds one of their four. They have the four receptor H1, H2, H3, H4. Okay. These receptors have different tissue distribution and mediate different effects on histamine binding. Serotonin, that is the is also present in mast cells of rodent and has effects similar to histamine. Okay, for example, if the histamine binds to H1 receptor, okay, this all are the receptor H1, H2, H3, H4 which are present on tissue cells. Okay, so suppose histamine binds to the H1 receptor. Okay. So allergic reaction are mediated, okay, by the binding of histamine to H1 receptor. This binding, okay, H1 receptor, histamine binds to the H1 receptor. This binding induce the contraction of intestinal and bronchial smooth muscles. Okay, it is the smooth muscles contraction, this function, and increase the permeability of venules. Okay, that is the small veins, and increase mucus secretion. The histamine binds to the H1 receptor. Or if the histamine binds with the H2 receptor, so increase the vasopermeability, okay, due to the contraction of endothelial cells, and vasodilation, that is the relaxation of smooth muscles of blood vessels. Now there is also they stimulate the exocrine glands and increase the release of acid into the stomach. Okay. So there is a different between the function if the histamine binds to H1, H2, okay, and another, that is the binding of histamine to H2 receptor of mast cell, which receptor present on the mast cell, and another cell, that is the basophils. So if histamine binds to H2 receptor which are present on mast cell and basophils, they suppress the degranulation, and thus histamine ultimately exerts negative feedback on the further release of mediators. Okay. So vascular permeability, okay, here it is the both, okay. So histamine and serotonin both have the same function. Okay, so vascular permeability refers to the ability of blood vessels, okay, blood vessels wall, to allow the passage of substances such as fluid, nutrient, and white blood cells. Okay. Smooth muscles contraction. Okay, smooth muscles, as we know, that is the involuntary, and is found in the walls of internal organs and blood vessels. Okay. Now, secondary mediators. Okay, first we finish the primary mediators. Both have the same function. Now eosinophil chemotactic factor. Okay, the function, it is the eosinophil chemotaxis. Okay. So eosinophil chemotaxis refers to the movement or migration of eosinophil in response to chemical signal. Okay, eoxin. Suppose they produce eoxin, that one of the chemical. Okay, the movement and migration of eosinophils. Okay, is likewise neutrophil chemotactic factor, NCF. Okay, here it also refers to the movement or migration of neutrophils in response to the chemical signal. Proteases. Okay, so proteases mediators, okay, primary mediators, tryptase or chymase. That function or effect, it is the bronchial mucus secretion. Okay, first degradation of blood vessels basement membrane, and generation of complement split products. So the complement splits product, okay. So as we know, the complement system, okay, is a part of the immune system that consists of group of protein that work together to defend the body against infection. Now the complement's activation can lead to the generation of various complement split products, which are fragments resulting from the cleavage of complement protein. For example, activating C1 then cleaves the C4 and C2 into smaller fragments. Okay, for example, C4A, C4B, and C2 also fragmentized by the C1. Okay, so C2A and C2B. Okay, this is the how the generation of complement split products. They split the protein into different products. Now the secondary mediators, okay, include the platelet activating factor, leukotrienes, here it is leukotrienes, slow reactive substances, okay, prostaglandins, bradykinin, okay, and cytokine and chemokines. These all are the secondary mediators. Okay, so as secondary mediators, the leukotrienes and prostaglandins are not formed until the mast cell undergoes degranulation, and phospholipase signaling initiate the enzymatic breakdown of phospholipid in the plasma membrane. Okay, once the degranulation is really occurs, and then the secondary mediators are released from the cell. Its resulting enzymatic cascade generates the prostaglandins and leukotrienes. Now, what is the function of leukotrienes? Okay, so leukotrienes, here it is the increase of vascular permeability as well as contraction of pulmonary smooth muscles, Okay. So in type 1 hypersensitivity mediated asthmatic, okay, if we talk about the asthma, so the initial contraction of human bronchial and trachea smooth muscles is at first mediated by the histamine. Okay, first they do by the histamine. However, within 30 to 60 seconds, further contraction is signaled by the leukotrienes and prostaglandins. So prostaglandin, leukotrienes, further the contraction of pulmonary smooth muscles. Okay, both here. That is, here is the contraction of pulmonary smooth muscles within 30 to 60 seconds after the allergen exposure. Okay. Now the pneumomole. Okay. So leukotrienes concentration, suppose it is has nanomoles level, the leukotrienes approximately 1000 times more effective and mediative bronchoconstriction than in histamine. Okay, and they are also more potent stimulators of vascular permeability and mucus secretion. That is the most important. Okay, leukotrienes, the level is nanomole. It's very effective to contraction of pulmonary smooth muscles. Now in humans, leukotrienes are thought to contribute significantly to the prolonged bronchospasm. Okay, so bronchospasm refers to the sudden constriction or narrowing of the airways in the lungs. That is a bronchospasm. Okay, and here is the bradykines. Okay, so bradykines increase the vascular permeability, smooth muscles contraction. Okay, vasodilation, prostaglandin also vasodilation, platelet aggregation, platelet activity factor, that platelet aggregation and degranulation, and contraction of pulmonary smooth muscles. Okay. Now we talk about the cytokines. Yeah. So adding to the complexity of the type 1 reaction is the variety of cytokine released from the mast cell and basophils. So suppose mast cell, basophils and eosinophil. Okay. So three major cells are responsible for the primary and secondary mediator production. So for example, they release the interleukin, okay, including 4, 13, 1, 3, 5, 6, 10. Okay, these are the interleukin, and also the GM-CSF and TNF alpha. Okay, these all are the released by the mast cell, basophils and eosinophils. These cytokines alter the local microenvironment where the infection occurs and leads to the recruitment of inflammatory cells such as the neutrophils and eosinophils. For instance, interleukin 4 and interleukin 13 stimulate the Th2 cells. Okay, they stimulate the Th2 cells. Okay, so Th cells response and thus increase the Ig production by the B cells. So here is the increase the Ig production. Now interleukin 5, this function, okay, especially important in the recruitment and activation eosinophils. So that is the eosinophils activation and recruitment. Okay, particular. The high concentration of tumor necrosis factor alpha secreted by the mast cell may contribute to shock in systematic anaphylaxis. Okay, so this is systematic anaphylaxis. So what is the anaphylaxis? Is a severe and potential life-threatening allergic reaction that occurs rapidly after exposure to an allergen. Okay, so this type of allergic reaction is systematic, meaning it affects in the entire body. For example, skin reaction, itching, flushed or pale skin, respiratory symptoms, difficulty breathing, wheezing or swelling of the throat and tongue. These are the systematic anaphylaxis. Another third, that is the cardiovascular symptoms, also including the rapid or weak pulse, drop in blood pressure, dizziness and fainting. And another, that is the gastrointestinal symptoms, nausea, vomiting, diarrhea and abdominal pain. Okay, so these are all the systematic anaphylaxis. Now the interleukin 8, okay, which are not mentioned here, interleukin 8 act is a chemotactic factor. Okay, this is a chemotactic factor and attract further neutrophils, okay, various neutrophils, basophils and various subset of T cells to the site of hypersensitive reaction. Now we talk about the GM-CSF. that is the granulocytes, macrophages, colony stimulating factor. They stimulate the production and activation of myeloid cell, that is white blood cells. Okay, granulocytes, basophils, eosinophils. Okay, both all are the granulocytes and macrophages. Okay, so and another, that is the function interleukin 1 and TNF alpha. Okay, so increase the expression of adhesion molecules on granular endothelial cells. So adhesion molecules facilitate the interaction between circulatory immune cells such as the leukocyte and endothelial cells lining blood vessels. Okay, so these increase expression of adhesion molecules like selectin, integrin. Okay, what are the and allowing immune cells to exit the bloodstream and migrate into tissue in response to infection or injury. Okay, where the infection injury happens, okay, they allow to go cells like eosinophil, basophils from the bloodstream. Okay, so this is the all the principal mediators involved in the type 1 hypersensitivity. Okay, there are the primary and these are the secondary, and these effects we can also say the function. Okay, okay, now the MCQ based question. So first, which of the following is considered a primary mediator in hypersensitive reaction? Okay, this leukotrienes is secondary, interleukin secondary, which are not eligible. Okay, histamine. Answer histamine. Primary mediators are released directly from the which cells release the direct primary mediators. So this is a mast cells. What is the primary function of histamine in hypersensitive reaction? Okay, vasodilation. Okay, answer is this B. Okay, which is interleukin function. Okay, stimulate antibody production, inhibit inflammation. This is no any function of any primary or secondary mediators. Okay, so okay, so for it is the leukotrienes are secondary mediators. They play role in bronchodilation, no, vasodilation, no, smooth muscles contraction. Okay, leukotrienes. Which of the following not secondary mediator in hypersensitive reaction? Prostaglandins, yes. Platelet, yes. Chemokine, yes. But interferon, which is produced by the infected virus infected cells. So this is the answer is correct, C. Okay. What is the primary source of cytokines in hypersensitive reaction? Okay, cytokine primary source, that is the mast cells, obviously. Okay, so for 7 question, it is platelet acting factor is secondary mediator involved in. Okay, inhibiting blood clotting, no. Enhancing platelet aggregation. Chemokines released in hypersensitive reaction are responsible for chemokine. Okay, so attracting immune cells to the site of inflammation. Answer is B. In the late phase reaction hypersensitivity, which secondary mediators contribute to the extensive tissue damage? Yes, so that is the leukotriene. Answer is leukotriene. Which immunoglobulin is directly involved in the activation of mast cells, leading to the release of primary and secondary mediator by hypersensitivity reaction? Okay, answer is IgE. Okay, so mainly hypersensitivity 1, IgE antibody, which are responsible for the release of primary and secondary mediators. Okay, so this is all about today's video. I hope this will help you. Thank you for watching.